Hello everyone, it's Fiaz and I'm going to discuss an article published in Nature Cardiovascular Research, and its title is Lipid-associated macrophages transition to an inflammatory state in human atherosclerosis, increasing the risk of cerebrovascular complications. The background of this study is that because carotid artery disease is the main disease related to cerebrovascular complications, and during this disease, the plaque formation happens in the blood vessels, or the fatty acid starts accumulating in the blood vessels, results in the obstructive or the hindered blood flow towards the brain. Here in this figure, we can see that the fatty acids are accumulated in the blood vessels, and after the plaque formation, number of the immune cells move towards this inflammatory site. Now, the main focus of this study is basically the macrophages and its two subsets, that are the lipid-associated macrophages. These two classes of the lipid-associated macrophages are TREM2, and it is a triggering receptor expressed on myeloid cells too. And the main role of TREM2 class of lipid-associated macrophages is that these are basically involved in the lipid uptake, and these enhance the lysosomal metabolism, antioxidative function, while they have the reduced number of the inflammatory genes. The second class of these lipid-associated macrophages is the newly identified community, which is the combination of FLIN2 and TREM1. And this class basically have increased lipid loading and the inflammation role, and they also exhibit a unique chemokine signature. So the objectives of this research or this study were the identification of selective targeting of intraplaque lipid-driven inflammatory pathway and phenotype transition rather than generic immune modulation strategies. Secondly, the second objective was to highlight new avenues for the treatment of cardiovascular diseases and other cerebrovascular complications. The results section, the first figure of the results is about the identification of the plaque T and natural killer cell populations with cytotoxic and activation signature. For the identification of these T cells and the natural killer cells, the researchers basically recruit 6 patients, and they extracted their carotid plaque from the carotid artery. And after the extraction or the sorting out of the CD45 cells, they observed that as the CD45 cells are mainly composed of the T cells and the mononuclear phagocytes. So further, they analyze the heterogeneity of these T cells and the mononuclear phagocytes. In case of the T cells and the natural killer cells, they found 11 clusters, and this UMAP graph is basically showing all those clusters or all those classes that they identified, and each color represents a single class of the T cell. In the second, in the B graph, there are the various signatures or the gene markers that are basically involved in the inflammation, and these genetic markers are present in these T cells. The color basically shows the expression level of these markers in these different clusters. In the bottom graph, this is basically a dot plot, and here along the y-axis, there are the clusters of the 11 clusters that they identified in the plaque, in this carotid plaque, and along the x-axis there are the genetic markers. The size of each dot is basically representing the number of the cells present in the clusters, and the color of and the color variation in the color is basically representing the average expression level of each signature present in each class. For example, if we see that along x-axis there is a signature marker that is CD3D, it is present in the proliferating or the CD4 T cells, and the color shows the moderate expression of these markers in these cells. While in case of CD8A, if we see that there is a maximum expression of this signature marker in CD8 T effector cells. In continuation, this is again a dot plot, and in this graph, along y-axis, there are the number of the pathways that are basically taken from different databases. As the color along the y-axis, you can see that the green color is basically those representing those pathways taken from gene ontology biological process. The orange color pathways are taken from the CAG, and similarly from the other one is from gene ontology molecular functions. Similar to the previous graph, the size of the dot is showing the number of the cells, and the color of the dot is representing the association or the odds ratio of each cluster with its respective pathway. In case of CD4T effector cells or effector modulator cells, if we see that this class of T cells is basically linked with showing an association with TH17 differentiation pathway, but this association is somehow moderate. While this CD4 T cells showing a strong association with TNF signaling pathway. So from this graph, we basically find out the linkage and the number of the specific number of specific cells present in the cluster and their association with the different pathways, inflammatory pathways and other pathways. After the heterogenetic analysis of the T cells, the second class that they identified in the CD45 were mononuclear phagocytes, and this result is about the plaque myeloid cells harbor diverse subset of the macrophages with distinct gene signature of the functional association. Now, in this part of the experiment, they focused on the mononuclear phagocytes, and they identified two main classes in the mononuclear phagocyte, the dendritic cells and the macrophages. And they found the 4 clusters of the dendritic cells and the 8 classes, the 8 macrophages populations. And this UMAP graph is again showing those 12 clusters, and each different color is representing a specific class of the macrophage and the dendritic cell. This UMAP graph is basically helping in visualizing and understanding the large dimensional data sets. And here we can see the main focus that we'll discuss in the coming slides, that is the TREM2 and the PLIN-TREM1. And this PLIN-TREM1 is newly identified community or the cluster in the plaque, carotid plaques. This B graph is showing again the different genetic markers that were found in these clusters, T cells, and again the color intensity showing the expression level of these genetic markers in these identified clusters. The lower in the lower graph of this slide, that is the dot plot. Now there is the correlation between, this tells us the correlation between the genetic markers along x-axis and the identified mononuclear phagocytes along the y-axis. And again, the cell size is represented by the cell number is represented by the size of the dot, and the color is showing the average expression of the specific marker in the respective clusters. Here AXL, that is a genetic marker, and we can see that this genetic marker is showing maximum expression as it is, and as the red color indicates, in the dendritic cells, while this marker is showing the least expression in the macrophages. Now, in continuation of this second part of the experiment, this is again the dot plot, And here the pathways that were extracted from different databases, and we have the markers, we have the clusters along the cells, clusters along the x-axis. Here, if we see that this S100A8, this specific cluster of macrophages, this is basically showing the association with different pathways, but the size of the circle is showing the number of these clusters or the cells present or linked with this specific pathway, that is the complement and coagulation cascade, while the color is showing that this association is very mild or very less. While if you can see that here is the red circle, so mean there are the number of the mean increased number of the class these cluster as 100A8, and these are significantly associated or linked with IL-17 signaling pathway and TNF signaling pathway, and the rest of the clusters we can see their associations and the numbers. After the heterogeneity analysis of the T cells and the macrophages, as they perform the trajectory analysis of the plaque macrophage populations, and through this trajectory analysis, they basically perform the RNA velocity analysis they conducted, and using the RNA velocity analysis, we can predict the future state of any individual cell. And here, this UMAP graph in the panel A, this UMAP graph is showing the conversion of or the transition of one phenotype or one cluster into the another. The main focus of this study was to observe the transition of this TREM2 into a newly identified community, that is the combination of PLIN2 to TREM1. This can be better visualized in the E panel, and in this panel, the direction of the arrow is showing the transition of TREM2 into the newly identified community PLIN2 TREM1, and the thickness of the arrow is showing the abundance, that how much of the TREM2 cluster is converted or transited into this newly identified community. Later on, they perform using they calculated the AU cell score in order to find out the genes that are identified or that were present in these clusters, especially this PLIN2 TREM1, and they considered those genes that were expressed in at least 10% of the cells. So in this B graph, this UMAP is showing the presence of those signatures that are lipid associated, and this AU cell score is showing that the color from the AU cell score is showing the presence of these specific lipid signatures. As the color is yellow or green, it means there is the presence of these specific lipid signature markers. Similar to this one, uh, here the apoptotic signature markers were visualized using this UMAP score, and they also found some inflammatory signature markers that is represented by this yellow and orange color in the PLIN2 TREM1 cluster. Along with this, they also performed the cytotrace analysis, and in this network, it can be visualized that there are increased number of yellow dots, as it depicts the presence of the lipid-associated macrophages or the genetic markers that enhance the accumulation of the lipids and that reduces the cholesterol efflux. So this can also help to differentiate, in order to differentiate between the TREM2 and the other community, that is PLIN2 TREM1. After this, now in this G panel or G graph, this is the volcano plot. And this volcano plot is showing the presence of the different gene markers or the signatures present in both the clusters, that is TREM2 and the other one is PLIN2 TREM1. And here it can be seen that this PLIN2 TREM1 is basically abundant with with those proteins that are involved in the increased inflammation and the reduced cholesterol efflux, and there are some of the chemokines, unique chemokines like VCAN, IL-1B, CCL2. So these are the chemokines that were found in this newly identified community, but these were not present in the other macrophage cluster, the lipid-associated macrophage TREM2. Moreover, this reddish area is also showing the abundant expression of PLIN2 and TREM1, and these are also involved in the increased inflammatory state at the plaque site. On the right side of this volcano plot, again the various genes or the markers that are abundant in the TREM2. These are present in this site. And here we can see that this reddish area is showing the abundance of those genetic gene markers or the signatures that are involved in the reduced inflammation, like NR1, H3, LIPA, and X2. These are basically involved in the in the increased lipid metabolism, or they increase the lysosomal degradation, and they also improve the cholesterol efflux. They help in the improved cholesterol efflux. After this, this graph showing in the panel H, this graph is showing the number of the genes present and their association with the different pathways. So again, the size of the circle is showing the number of the genes, and the color of the circle is showing the significant association and the significant association of these genes with the specific pathway. Here in the upper part, it can be observed that these genes are lying on the positive in the positive quadrant, and they are showing the positive association and with the respective pathways. While, for example, we can see that here this is the gene that is showing the positive association with endocytosis BP, but this association is not significant. It is significant but less as compared with others. While in the lower this panel, there are the number of the genes that are lying in the negative quadrant. So they are showing that they are basically down regulating, or they have the negative association with these respective pathways. And again, the size and the color is showing the number of the genes and the significant association with the respective pathways. Later on, this study also added that, the researchers also performed the histology of by taking a tissue from the plaque, and they took their stenosis region, almost 1 mm of the tissue, and they performed the immunohistology. And after immunostaining, they found they traced the location of different proteins like PLIN2, TREM1, and TREM2 as compared with the controls. Like the red is showing the PLIN2, the red arrow is showing the location of, and similarly this blue one is showing the location of the TREM2, while this black one is showing the location of all of these complexes together in the plaque area. Moving to the next part of the experiment, and here they tried to find out the cell-to-cell interaction analysis. They performed the cell-to-cell interaction analysis that suggests a central role of macrophages in the immune cell communication network of the human atherosclerotic plaque. Here are the two heat map graphs, and using the network analysis tool and multicellular interaction, they basically draw these heat maps. And from in the first graph A, it can be seen that in this graph is basically showing the cell-to-cell interaction. So the color of each box is showing the specificity of the cell-to-cell interaction, while the number shows the the number shows the significant association of and ligand receptor interaction. So those which are significantly linked, those ligand receptors which are significantly linked are given here. As we can see that the maximum specificity was found in case of the max cell, following by the CDC, and this specificity is significant also. Then we can also observe this specificity using this heat map graph, relation between the ligand receptor connectivity and its respective cell. As I was discussing that in case of the MAC inter-cell to cell interaction, we can see that there is a maximum number of receptor ligand interactions, and these interactions are highly specific, as well as we can see from this scale score that as the color gets darker, so the specificity gets the interaction is getting more specific. So in case of MAC to MAC cell interaction, the least ligand to receptor interaction has been observed in case of this combination. So after this, they also performed an experiment to check the relative gene expression of various proteins found in the macrophages at the plaque area, and where they exposed these genes against various ligands, and those ligands are differentially I mean, these ligands are basically represented by different color codes, and here in these box plots, we can see the relative expression of all these these genes, and after exposing against different ligands. In case of the TREM1, we can see that this TREM1 is highly expressed against ACCM ligand, and while the TREM2 is highly expressed against oxidized LDL ligand. Similarly, the other graphs are also showing the maximum expression against different ligands. So after that, they also performed an experiment to check the effect of toll-like receptors on the phenotypic transition from TREM2 to the newly identified community or cluster, that is the combination of PLIN2 to TREM1. And for this purpose, they basically silenced or knocked out the toll-like receptor 2 gene, and after knocking out, again they exposed the cells against different ligands, and these color codes are showing the different ligands that they used against these proteins. So in the first graph, like in this graph K, we can see that after exposing, in case of the control, after exposing, in case of control, after exposing against the FSL and ACCM ligand, the TLR2 has showing the maximum expression against ACCM, while in case of the knocked out toll-like receptors, there is no expression of this specific protein. Similarly, it can be observed in the graph L and M, that after the knocking out of toll-like receptor 2, even the expression of the TREM1 and PLIN2 is significantly decreased after exposing against the same ACCM and FSL ligands that they used before as well. But here the TREM1 is showing the maximum expression against this ACCM, while in during the after the knocking out of this toll-like receptor, this the expression of the TREM1 and PLIN2 is significantly reduced. Uh, mm, they mean that the researchers also validated their study after performing another experiment, and for this experiment they basically took the patients from CPIP biobank, and they categorized the patients into symptomatic and asymptomatic. They considered those patients who came symptomatic, who came for their surgery, and the stenosis rate was greater than 70%. Moreover, they have some symptoms as well, like blurred vision and stroke ischemic and transient ischemic stroke. While those were they considered asymptomatic, who were not showing any symptoms, but the stenosis was greater than 80%. So they took their carotid plaque samples, and after the extraction of the various immune cells, they created the RNA library, a single cell RNA sequence library, and they also performed their histological analysis as well. So in this B, this B is showing is basically showing the histological picture of and these carotid tissues that were obtained from the plaque area and the normal area. And here it can be seen that there is a clear difference between the normal and the carotid tissues obtained from the plaque area. Similarly, this one is showing the immunostaining picture of PLIN2, and this one is for TREM1, and it can be clearly differentiated from the control. Moreover, they also try to find the presence of the TREM1 and PLIN2 in terms of its total plaque area. And this graph is showing that there is a positive linear correlation between the plaque area of TREM1 and the PLIN2. Similarly, in the D graph, this D graph is showing the gene expression of the TREM1 and PLIN2, and this is also showing the linear and the positive correlation between these two proteins. Furthermore, these graphs, these box plots are also showing the gene expressions of the various gene markers, in case of E and F box plot, the green color is showing the symptomatic patients, and the blue color is showing the asymptomatic. And for the expression level of the PLIN2 and TREM1, it can be clearly seen that the expression level of PLIN2 and TREM1 are significantly higher in the symptomatic patients. And similarly, the total plaque area of PLIN2 and the TREM1 was also higher in the symptomatic patients. Similarly, they also tried to find the gene expression of various gene markers present in the PLIN2 TREM1 community. And again, here the color of the box plot is representing that the orange color is showing the high PLIN2 to TREM1 cell ratio, and the green color is showing that cell basically consists of a low number of PLIN2 to TREM1. So again, the clear and the significant difference can be seen in terms of the gene expression for toll-like receptor 2, 4, and the other gene markers. that these gene markers were significantly higher in those plaque area or the in those plaque area in which there were a high ratio of plin 2 to tram1. So this plin 2 to tram1 is basically involved in the increased in the inflammation state. So the take-home message that, as per my understanding, the take-home message from this research study is that there is a transition between the two between the two clusters of the lipid-associated macrophages. That in case of more inflammation, when the TREM2 cluster is transited or is converted into the newly identified cluster, that is plin2 TREM1. So this basically enhances the inflammatory state. So this plin2 TREM1 cluster reduces the cholesterol metabolism and efflux, while it increases the inflammation and the specific chemokines to the plaque area that further make the disease worsen and cause more disease complications. The one more result or the conclusion from this study is that the toll-like receptor 2s, the toll-like receptor 2 might be involved in the increased expression of these inflammatory gene signatures that are the plin2, TREM1, IL1B, and CCL2. Hope all of you have got what I tried to explain. See you in the meeting on Tuesday. Bye.